So what do I do?
What are you doing?
What do I do? Do I tell you what I'm doing?
These 4 days are dedicated to the IAs in science for group 4. I don't know, mister, honestly.
So I am measuring the transpiration rates in plants and the effect of temperature, wind, and humidity that it has on it. And I'm doing that by measuring the initial mass of the plant and then the final mass after a certain amount of time. And it should, according to my hypothesis, it should decrease as the temperature increases. It's a humidity meter to measure the humidity. So I don't know if you can really see it.
Okay.
Yeah, I put it like in there and then it shows me the humidity of the plants. And then this is a light meter, which shows me the light intensity.
Keep it. We grew bacteria and we're testing different antibacterial substances on it. So he was testing different toothpaste and different mouthwashes, and I was testing different ingredients of hand sanitizer to determine which ingredient in hand sanitizer is the antibacterial one.
So this is a light box, and these are the petri dishes in which we grew the bacteria, and then yesterday we added the antibacterial substances and we left them overnight in the incubator. So now we're using this sheet of graph paper to count how many squares are covered by bacteria in order to determine which substance killed most bacteria. She actually wants to be a scientist though. So.
Just take a little piece of root, put it on a slide, put it a cover slip, and we'll have a look at it just like that. And this, keep it, don't throw it away, label it, and maybe we're going to use that later too.
So I was told that there's a student back here in the bowels of the school. I've never actually been back here, kind of behind the science labs in the basement, but apparently she's back here doing an experiment, playing with light. Let's find out more.
So this is an aquatic plant. I take it, I cut it, and I put it in the graduated cylinder. I also add sodium carbonate to it in order to increase the concentration of carbon dioxide. So then we can measure the oxygen, and we measure the oxygen through oxygen bubbles that are formed because of photosynthesis. So the higher the amount of oxygen bubbles, the higher the rate of photosynthesis. I've noticed that colors like blue and red actually increase the rate of photosynthesis, and green, because the plant leaves have chlorophyll, it reflects the light, so basically I'm pretty sure that's what decreases the rate of photosynthesis.
So basically yesterday, I tried my original experiment for the entire day, basically. And what my original experiment was I would take like these leaf discs. I would cut circles out of leaves, specifically basil leaves, and I was going to take the oxygen. I did take the oxygen out of them with a syringe by creating a light vacuum to take out all that oxygen, and all the leaves then sunk. But the problem was they weren't floating. So I changed pretty much everything I could think of. I changed the carbon dioxide concentration, the way that I was cutting the leaf discs, the light source, the light intensity. I even tried some outside and it wasn't working. And the conclusion that I've come to is that the problem was that the leaves were too young. So I did some pre-trials at home where I used basil leaves from the supermarket, so they were just bought, they were already ready for cooking. But what I did for my IAs is I went and I bought a basil plant from the supermarket instead of actual basil leaves. So that's where I think it went wrong. But then today I'm going a different path, and I'm using this oximeter, which basically measures the concentration of oxygen in water. So I'm using a different plant, an aquatic plant. And I'm measuring it at different light intensity.
Doing some a reaction using fermentation in glucose with glucose and yeast, and I'm checking the carbon dioxide production now, every 30 seconds. This is glucose and yeast together with distilled water, and these are the other sugars because I'm testing in different types of sugar how the reaction is going to occur. This is lactose, sucrose, fructose, and maltose. This was the glucose.
For me, I love doing science, so just having 2 days to just spend in the lab, or 4 days for me because I also do physics, it's been a lot of fun. There's been a couple challenges that have popped up that were unexpected, but half the fun is trying to figure out how to work around those to make sure you get a good result or a result that you can write a report on anyways.